This is the article I chose. It is named Differential Expression of Wilms Tumor Protein in Diffuse Strength Protein Glioma. So let's start talking about some generalities about the topic. So what is this diffused in strength protein glioma? It's a very fast-growing tumor that it's located on the glial cells of the pons on the brain, and it spreads very, very fast to adjacent tissues. So that makes it a very hard to treat tumor, and it also gives patients a very poor prognosis, as it is affecting one of the most important parts of our brain. It usually occurs in children, and it do not discriminate sex. It occurs equally on boys and in girls. And although it is a very rare tumor, each year in the United States they diagnosed like up to 300 new cases of these disease. So actually, these investigation could impact an important population. So some of the signs and symptoms of a patient that could have this disease include everything that has to be like with the main nervous system control, but generally it includes like problems with eye movement, problems of hearing, troubles chewing or swallowing, like very simple things that we're used to do without having a challenge with it. They also have like very weak limbs, so they have difficulties standing or walking, and as the tumor is located on their hands, they have very hard headaches. They could have brain edemas, and it's very common for these patients to have nausea or constant vomiting. So then these Wilms tumor protein, we have to know that it's encoded by the WT1 gene on chromosome 11. What is it? So it's a transcription factor which regulates many cellular development and cell survival aspects, but it is known for specific special things such as the regulation it has on the expression of numerous target genes, such as EPO, or the importance it has on the development of urogenital system of people, and it is known as a highly associated with as a protein highly associated with various cancers. And actually it was ranked by the National Cancer Institute as the #1 target for cancer immunotherapy. And in some trials they have been trying this vaccine, that what it does is that it induces an acquired an immunity for these, like, against this protein. Okay, so the objective of the investigation was obviously to find out the differential expression of this specific protein in this specific tumor. So they used a couple of methods, but I chose some of them to explain it to you. So the first one is quantitative RT-PCR. This RT stands up for reverse transcription and not for real time. You guys don't confuse them both, because they are written the same way, but they're different methods, so you have to read very good and search which one of them is it. So what they do with this method, it's that they do an amplification of RNA through its copy DNA, synthesized a priori with using reverse transcriptase, but it's an enzyme, and this way they can follow several cell cycles and actually determine the expression of some specific genes in these various tissues and various processes the cell passes through. So in this experiment, this investigation, they use it using a copied DNA from frozen tumor specimen, and they confirmed, they took some samples of tumor tissue and some samples of adjacent normal tissue in order to have something to compare with. So with this one, they found out that the expression of the protein was significantly higher in cells that had been taken away from the tumor sector and not in the, rather than the adjacent tissue that they took. The immunohistochemistry, actually, um, everything that has immune on it has to do with antigens and antibodies. So what they did here, it consists on using antibodies to check for certain antigens, but in a specific sample of tissue. So the antigens, the antibodies are usually linked to an enzyme or to a marker like a dye fluorescent marker or something like that. And after the antibody binds to the antigen, they can be seen under the microscope in these sample tissue. So actually, in this investigation, what they found it out with then when they did these immunohistochemistry, was that they associated these transcription factor with the expression of the mutated histone, the oncohistone H3. And as well as in the past method, they find out that the expression of the protein was higher in the samples of tissues that were taken out from the tumors region than the adjacent tumors tissue. Another one is the Western blot assays. This Western blot consists on identifying specific proteins from a complex mixture of proteins extracted from cells. And these techniques usually is organized in three simple steps or elements. I don't know how to say that, but yeah, it consists of three main aspects. So the first one is that they separate proteins by size, then they transport it to a solid separate, and by third they mark the protein target using an antibody that they can visualize. So what they did in this research was that they used a protein from the DIPG subjects from whose cells were available and normal tissue as well, and they showed significant overexpression of these WT1 and the DIPG specimens compared with the adjacent samples, as well as in the other past two methods. And as well with these Western blot assay, they found out that these cells, these how do I say that? These tumor cells were not the only ones that showed an overexpression of these protein, because as well other cells that had to do with central nervous system tumors in pediatric patients had it overexpressed. The last method I'm going to explain to you is the immunofluorescence. It's based on the identification of the reaction between a specific antigen, and an antibody. But what they do is that the reaction is targeted with the fluorescent reagent, which evidences not only the reaction itself but its intention. So the intensity, not intention. So the intensity of the fluorescent dye, it's directly proportional to the intensity of the reaction. So what they did here is that they used some antibodies against this specific protein, and they probed for mutant histones, the ones that I talked to you before, and they corroborated with the conclusion that stated that these transcription factor, the WT1 expression, is associated with the mutation status of oncohistone H3 that I had mentioned before. And as well, they could find out that this protein was located in the cytoplasm of the cells. Then this was a surprising finding out, because this protein has been known to be a nuclear protein. So yeah, now I'm going to explain some results This first table we are seeing here was a Western blot one. It actually, what it is explaining is that here the relative transcription factor signal intensity, and these numbers right here are just the patient numbers, like the patient's ID. And this T means for tumor and this N means for normal. So this the factor. You can see that it appears in every tumor. Oh my god, I'm sorry. In every tumor cell. This GIPDH was just the enzyme that they tested as well. But you can see that all the tumor cells have the factor present. Although in some of them you can't really like see them clearly, but there it is. This one was immunohistochemistry one. What we matter about here is just this one up here. And as you can see here, these are adjacent normal cells, and here are other midline gliomas, but this one are the DIPG. So they proved like various variants of the oncohistones, and they find out, I mean, that the overexpression that this protein had on tumor cells compared with healthy cells, you know. So this one's right here. This one was a result from one of the Western blot method, and this one was immunofluorescence. So this one is just showing up that the wild tumor transcription factor is present and overexpressed in every tumor cell, as well as the enzyme it was linked to. And here they're trying to compare with this one because this K562 was one of the cells where you could find that the real tumor protein was a nuclear protein. Here in the images, I don't know if you can get to see them very well, but here you can visualize that the protein is expressed on the nucleus, and in the other images you can see them on the cytoplasm. That was the one that I told you that was a very surprising finding out. About the discussion, I took like three phrases that authors said and analyzed if they matched with the article. And actually, the only one that didn't match was this one, because it said that our systolic detection of WT1 is not surprising, as previous studies have also shown systolic localization of the factor in glioblastoma. I mean, these do not matches with the article, as in the article they state that this was a very surprising find out, as the protein is, it's known for being a nuclear protein. And then the other two, oh, I'm sorry, you can see very good this one, but the other two did match with the article, what with the article said. So in conclusion, I think the article was very well performed, and as I told you at the beginning, it's going to have probably a good impact on a not very big but in an important population. And as I acknowledge the advances that genetic therapy has been having, like nowadays, and since a long time ago now. Some cure or some treatment could be found for this disease and cure various pediatric patients. And the other one is that it's stands up the molecular biology importance, as it enhances the value of knowing the origin of the diseases from the very simplest and smallest factors, which can fully help like medical or medicine in general to improve not just these but other patients' qualities of lives. And that's it. Thank you very much.